Welcome back to the deep dive. Today we're not just talking about your standard infections. We're talking about a home invasion.
Yeah, but not the kind where they just kick down the front door.
Right, exactly. We are looking at the stealth operatives of the bacterial world, the ones that don't just enter your body, they enter your cells. They basically look at your immune system, laugh, and then set up a living room inside the very structures that are designed to kill them.
It's a terrifyingly elegant strategy, honestly.
Mm-hmm.
Usually when we talk about bacteria, we think of them as these little soldiers marching around in the blood or the tissue fluids, fighting a war on an open field.
Like strep or staph.
Exactly. But the organisms we're discussing today, Rickettsia, Ehrlichia, and their cousins, they've decided the safest place to be is behind enemy lines. Inside the bunker.
Inside the bunker. I love that. So for everyone listening, we are doing a comprehensive deep dive into Chapter 34 of Murray's Medical Microbiology. That's the primary structural spine for today.
Right, the chapter covering Rickettsia, Ehrlichia, and related bacteria. And we're supplementing a bit with Gillette's Chapter 26, but Murray is really our roadmap here.
And look, I know for a lot of you medical students, people studying for the USMLE, or just curious minds, this chapter feels like a graveyard of names that all sound exactly the same.
It really does. You've got Rickettsia, Aurientia, Anaplasma, Coxiella. It's just a mess of Latin names and bug bites.
It is the vector-borne confusion chapter. So our mission today is to dismantle that confusion. We are going to take that laundry list and turn it into a structured mental map for you.
Because we aren't just memorizing names today. We are going to understand the personality of each of these bacteria. Because once you understand how they survive, the disease they cause makes perfect sense.
So we're building a spine for this deep dive. We're going to move from the Rickettsia, which are the ones that cause your blood vessels to leak, to the Anaplasmatacea, the ones that hide inside your white blood cells. And then we'll finish with the ultimate survivalist of the group, Coxiella.
And if there is one thematic anchor, just one big picture concept I want you to hold on to for this entire deep dive, it is this. Location, location, location.
You sound like a real estate agent.
In microbiology, it's remarkably similar. Because unlike many gram-negative bacteria that wreak havoc by pumping out exotoxins,
like cholera or something.
Yeah, Vibrio cholerae or certain strains of E. coli, these organisms we're talking about today generally do not produce toxins. They don't need to.
Wow.
They cause disease, primarily by where they choose to live inside the cell and how your immune system panics in response to that intrusion. Where they hide determines the pathology.
So no toxins, just really, really bad roommates.
Exactly.
Let's start with Section 1, General Taxonomy and Physiology. This is the setup. Historically, I feel like those were all just thrown into one bucket, right? Like the weird small bacteria bucket.
They were. For a long time, if it was small, lived inside a cell, and you got it from a bug bite, we just called it Rickettsia. It was a catch-all term.
Because they look similar under the microscope.
Right. They looked similar under the light microscope, these tiny little rods, and they were all obligate intracellular parasites. But then came the genetic revolution.
16S ribosomal RNA sequencing ruins the family reunion.
It really forced a divorce, essentially. Modern DNA analysis showed us that despite their similar lifestyles, they are genetically quite distinct. So now we have a used-to-be family situation. There are 3 distinct families you need to know for the boards.
Okay, lay out the custody arrangement for us.
First, you have the Rickettsia. This keeps the namesake genus Rickettsia and adds a cousin called Aurientia. These are your vasculitis makers.
Okay, Rickettsia and Aurientia. Got it.
Second, we have the Anaplasmatacea. This family contains Ehrlichia and Anaplasma. These are the ones that invade blood cells.
Blood cell invaders.
And finally, standing largely on its own in the corner, is Coxiella, containing Coxiella.
And isn't Coxiella actually related to Legionella? I remember reading that somewhere.
It is. Genetically, Coxiella is much closer to Legionella pneumophila, the cause of Legionnaire's disease, than it is to Rickettsia.
That's why.
But because Coxiella lives inside cells and was historically grouped here, we still teach them together. It's a marriage of convenience for the textbook writers.
Okay, so 3 families. But they do share some common traits. Otherwise, we wouldn't be doing one massive deep dive on them. Let's talk about their appearance. Murray calls them gram-negative, but then immediately puts an asterisk next to it. What is the deal with that?
This is a classic exam trap. Structurally, they're gram-negative. If you looked at their cell wall under an electron microscope, you would see the inner membrane, the thin peptidoglycan layer, and the outer membrane with lipopolysaccharide, LPS.
So they have the ID card of a gram-negative bacterium.
Right. But if you actually try to perform a gram stain in the lab, you won't see much of anything. They stain very poorly.
Why is that? Are they just too small?
Part of it is size, they are tiny, but part of it is that they're intracellular. You're trying to stain a bacterium inside another cell, and the contrast just isn't there.
No, that makes sense.
Also, Ehrlichia specifically lacks peptidoglycan entirely, so it structurally doesn't even hold the concept together.
Okay, so if a question stem on an exam says gram stain revealed gram-negative rods, I should probably be thinking about something else, like E. coli or Pseudomonas.
Exactly. If the question gives you a clear gram stain result, you usually aren't looking at Rickettsia. For these guys, you need special visualization. You're looking for keywords like Giemsa stain or Jimenez stain.
Giemsa.
Or, more likely in a modern hospital, direct fluorescent antibody tests. But for the boards, remember Giemsa.
Got it. Now let's get back to that location theme. Being obligate intracellular means they must be inside a cell to survive. They can't just grow on an agar plate like staph or strep.
Correct. They have lost the metabolic genes required to make their own energy or amino acids. They are metabolic cripples. They are entirely dependent on the host cell's ATP, their energy parasites.
So they have to get in. And usually a human cell doesn't just open the door for a bacterium. How do they break in?
It's a seduction, really. A multi-step invasion. First, they adhere. Rickettsia, for example, expresses a surface protein called OMPA, outer membrane protein A.
OMPA.
Yes. It binds to receptors on the host endothelial cell.
Okay, so they stick to the surface. Then what happens?
Then they pull a Trojan horse maneuver. They stimulate phagocytosis. They trick the host cell into thinking, hey, I should eat this.
But wait, endothelial cells aren't macrophages. They don't usually just eat things.
Exactly. The non-phagocytic cell rearranges its cytoskeleton to engulf the bacteria.
But usually being eaten by a cell is a death sentence. That's the whole point of phagocytosis. You get pulled into a bubble called a phagosome. That bubble fuses with a lysosome, and then you get bathed in acid and enzymes until you dissolve.
That is the standard cellular defense, the stomach of the cell. And this is where our three families diverge beautifully in their survival strategies. This is the first mental model you need to build today.
Okay, let's build it.
I call it the Great Escape, the bunker, and the acid lover.
I love a good heist analogy. Break this down for us.
Let's start with the Great Escape. This is the strategy of the Rickettsia, Rickettsia and Ehrlichia. As soon as they are engulfed into that phagosome, they detect the change in the environment.
So they know they're inside.
Right. And before the lysosome can fuse and pour in the acid, they produce a phospholipase enzyme.
Phospholipase, so they attack the membrane.
They dissolve the membrane of the phagosome. They literally melt the walls of the jail cell.
That is incredibly risky. If they produce too much phospholipase, wouldn't they dissolve the host cell membrane too and just kill their host before they can replicate?
It is a very delicate balance, but yes, they could.
Yeah.
But they dissolve just the phagosome membrane and escape right into the cytoplasm. Now they're free. They're swimming in the nutrient-rich cytosol, safe from the lysosomes. That is the jailbreak.
Okay, so Rickettsia busts out. Who chooses the bunker?
That would be Ehrlichia and Anaplasma. They don't have that phospholipase escape mechanism. They stay inside the phagosome.
So they're tracked.
But they secrete proteins that modify the phagosome membrane to prevent the lysosome from fusing.
Oh, so they lock the door.
They lock the door, reinforce the walls, and turn the phagosome into a luxury condo. They replicate happily inside this vacuole, completely safe from the host's enzymes. We technically call this a parasitophorous vacuole, but the bunker is easier to remember.
The bunker is definitely easier. Okay, so Rickettsia is free in the cytoplasm. Ehrlichia is locked in a safe room. What about Coxiella? You called it the acid lover.
Coxiella is the most hardcore of the group. It stays in the phagosome. It does not prevent lysosome fusion. It lets the lysosome fuse. It lets the acid pour in.
Why? That sounds like suicide.
Because Coxiella has evolved to require an acidic pH for its metabolism. It actually waits for the acid bath to turn on its replication machinery.
Are you serious?
Yes, it turns the host's primary weapon into his activation switch.
That is twisted. It's like an organism that only wakes up when you set it on fire.
It's brilliant adaptation. And it's exactly why Coxiella is so incredibly hard to kill, which we'll get to later.
Okay, so that's the entry in survival. Let's move to section 2 and focus specifically on the Rickettsia, the jailbreakers. These are the ones that cause vasculitis.
Right. Within the genus Rickettsia, we generally divide them into two major groups for clinical purposes. The spotted fever group, or SFG, and the typhus group, TG.
Spotted fever versus typhus. And even though they both escape into the cytoplasm, they behave differently once they're out there. I've heard this described as the rocket versus the bomb.
That is a very accurate visual. Let's look at the spotted fever group, typified by Rickettsia rickettsii. Once it's loose in the cytoplasm, it needs to spread. But it doesn't have flagella. It can't swim.
So how does it move?
It hijacks the host's cytoskeleton again.
It builds a motor.
It creates an actin tail. It polymerizes host actin filaments at one pole of the bacterium. Think of it like a speedboat engine, but made of the cell's own structural beams. It uses this actin rocket to propel itself through the cytoplasm.
So it's just surfing around inside the cell?
And not just surfing. It generates enough force to push into the cell membrane, create a protrusion, and push right into the neighboring cell.
So it spreads cell to cell without ever really having to expose itself to the extracellular space? It just drills through the walls?
Precisely. It's a stealthy lateral spread. Now contrast that with the typhus group like Rickettsia prowazekii. They lack the specific gene to polymerize actin effectively. They can't build the rocket.
So they're just stuck there?
They are stuck in the cytoplasm. So what do? They replicate and replicate and replicate until the cell is so packed with bacteria that it physically bursts.
That's the bomb.
Lysis. The cell explodes, releasing the bacteria into the bloodstream to find new cells.
Okay, rocket versus bomb. But regardless of how they exit, the damage they cause is based on the tissue they target. And for Rickettsia, the target is the endothelium.
This is the single most important concept for understanding the symptoms. These bacteria specifically bind to and infect endothelial cells, the delicate wallpaper lining your blood vessels.
So the infection is happening inside the walls of the pipes.
Correct. As the bacteria replicate, whether they are surfing through the walls or blowing them up, they damage the endothelial integrity. The cells die or detach.
So the pipes start leaking.
The pipes leak. Yeah. And this explains everything. Why do you get hypovolemia, low blood volume? Because plasma is leaking out of the vessels into the tissue. Why do you get low proteins, leaking? Why do you get organ failure? Because the perfusion pressure drops.
And the rash. Everyone always talks about the rash.
The rash is essentially thousands of tiny hemorrhages. It's blood leaking from damaged capillaries into the skin.
That's a very visceral image. It's not just inflammation like a mosquito bite. It's structural failure of the capillaries.
And I really want to reiterate, there are no exotoxins involved here. The damage is structural, caused by the bacterial replication, and crucially, the host's own immune response. Your CD8 cytotoxic T cells recognize the infected endothelial cells and kill them.
So your immune system is trying to stop the infection by destroying the lining of your own blood vessels.
It's a scorched earth policy, and it contributes significantly to the vasculitis.
That leads us perfectly into Section 3, the big one, Rocky Mountain Spotted Fever, or RMSF, caused by Rickettsia rickettsii.
This is the heavyweight champion of rickettsial diseases in the U.S. It is potentially fatal, rapid, and a massively high-yield topic for every medical board exam.
Let's set the scene with a clinical vignette. If you're an ER doctor, just walk through the door.
You're typically looking at a patient who looks toxic. They have the classic triad: fever, severe headache, and myalgia, which is muscle pain. The headache is often described as the worst of their life.
And they need a vector exposure. For RMSF, that vector is the hard tick.
Specifically dermacenter ticks. In the east and south, it's the dog tick, Dermacenter variabilis. In the Rockies, it's the wood tick, Dermacenter andersoni.
Now, there is a very specific wait time involved with ticks. I've always told people, if you find a tick crawling on you, you're fine. If it's attached but flat, you're probably fine. Why is that?
That is the reactivation phenomenon. This is fascinating physiology. The bacteria in the tick are in a state of stasis or dormancy because the tick is at ambient temperature. They are metabolically inactive.
They're basically asleep.
Right. When the tick latches onto a human and starts feeding, the warm blood meal does two things. It raises the temperature and it provides nutrients. This signals the bacteria to wake up, downregulate their stasis proteins, and upregulate their virulence factors. This process takes time.
How much time are we talking about?
Generally, at least 6 to 10 hours, often up to 24 hours.
Right.
So the tick must feed for a significant duration to actually transmit the infection. This is why daily tick checks are so incredibly effective. You can pull them off before the bacteria have had their coffee and woken up.
That is a great public health tip. Now let's talk about geography. The name is Rocky Mountain Spotted Fever, so naturally I assume I should look for a hiker in Denver.
You would think so, and you would be falling right into a classic trap. The disease was first identified in the Snake River Valley of Idaho, hence the name. But today, The vast majority of cases in the U.S. occur in the southeast and south central belt.
So where exactly are we talking?
North Carolina, Oklahoma, Arkansas, Tennessee, Missouri. If you see a question about a hiker in the Appalachian Mountains or the Ozarks, think RMSF. If they are in the Rockies, it's actually much less common now.
That is the geography trap. Now let's talk about the rash, because this is the key clinical differentiator.
The rash is iconic, but it's tricky because it's delayed. It usually doesn't appear until day 3 or later.
So early on, you just have a fever and a headache. This is exactly why it gets misdiagnosed as the flu.
Exactly. And if you wait for the rash to treat, you might have waited too long. But when the rash does appear, it has a centripetal pattern.
Break down centripetal for us.
It means moving toward the center. The rash starts on the wrists and ankles. Then it spreads up the arms and legs to the trunk.
Wrists and ankles first, then trunk. That's very distinct.
And here is the crucial detail. It involves the palms and soles.
Palms and soles. That is a short list in dermatology.
It is the syphilis-Coxsackie RMSF differential. If you see a rash on the palms and soles, you have to rule these out. Syphilis is usually chronic and painless. Coxsackie, or hand, foot, and mouth, is usually in kids with mouth sores. RMSF is an acute, life-threatening febrile illness.
And the rash changes character over time, doesn't it?
Yes, it starts as macules, flat pink spots that blanch when you press them. But as the vascular damage worsens, it progresses to petechiae, non-blanching purple spotted hemorrhages. That's the spotted and spotted fever. Once it becomes petechial, it means significant vessel damage has already occurred.
Let's get into diagnosis and treatment. This is a life-threatening condition. Mortality can be as high as 20 to 25% without treatment. Do we wait for the labs to come back?
Absolutely not. I cannot stress this enough. This is a clinical diagnosis. Serology, like the microimmunofluorescence test, or MIF, is the gold standard for confirmation. But antibodies take weeks to develop. You will not have a positive lab test in the first week of illness.
So if you suspect it, you just treat it.
You treat based on suspicion. If you wait for the lab confirmation, the patient may die.
And what is the drug of choice?
Doxycycline. It is the answer for almost everything in this chapter.
Now, I have to ask about the doxycycline in kids issue, because I remember in med school being told, don't give tetracyclines to kids under 8 because it stains their teeth gray.
That is the general rule for tetracyclines. The drug binds to calcium in developing enamel. However, the American Academy of Pediatrics and the CDC have revised this specifically for doxycycline and specifically for RMSF.
Why has it changed?
2 reasons. 1, newer data suggests that short courses of doxycycline, unlike the older tetracyclines, cause minimal to no staining. 2, and way more importantly, RMSF kills children faster than adults. The mortality risk far outweighs the cosmetic risk.
So it's teeth versus death.
Exactly. Doxycycline is the first-line treatment for everyone, regardless of age, for suspected RMSF. Do not use alternative antibiotics. They just don't work as well.
Clear enough. Let's move to section 4. Other Rickettsia. These are the lookalikes. Let's start with rickettsial pox.
Rickettsia acari. This is a fascinating bit of medical history. It was discovered in 1946 in New York City. People in a specific apartment complex in Queens were getting sick with a fever and a chickenpox-like rash.
And the vector here isn't a tick.
No, it's a mite, specifically Liponosoides sanguineus, which is a mite that lives on the house mouse.
So you have mice in the walls, mites on the mice, and then mites biting the humans.
Exactly. It's a disease of the urban environment.
What's the giveaway for rickettsial pox?
The eschar. This is a key term. An eschar is a dead piece of tissue that forms a black, crusty scab. At the site of the mite bite, you get a firm red papule that turns into a vesicle and then forms this painless black eschar.
And what about the rash?
The generalized rash is papulosecular. It looks exactly like chickenpox vesicles. That's why it's called rickettsial pox.
But chickenpox is a virus. This is a bacteria.
Correct. And unlike chickenpox, you have the eschar. Also, rickettsial pox is generally self-limiting. It's not fatal like RMSF.
I remember reading about a confusion with anthrax regarding the eschar.
Yes, during the 2001 bioterror scares. Cutaneous anthrax also causes a black eschar. Looks very similar. But the key difference is the rash. Araucaria causes the eschar plus a generalized body rash. Cutaneous anthrax is usually just the local eschar without a widespread rash.
That is a great differentiator. Eschar plus all-over rash equals rickettsial pox. Eschar plus no rash, you better check for anthrax. Let's move to the plague of history, epidemic typhus.
Rickettsia prowazekii. This organism has likely altered the course of human history more than any general or politician.
You're talking about Napoleon's retreat from Moscow.
Precisely. The Grande Armée wasn't just defeated by the Russian winter. It was decimated by typhus. This is a disease of war, famine, crowding, and squalor.
And the vector is the human body louse.
Pediculus humanus corporis. Not the head louse you get in kindergarten. This is the body louse that lives in clothing. It proliferates when people cannot wash or change clothes for weeks.
How does the louse transmit it? Does it bite you?
It bites you, but the transmission is actually grosser than that. The louse defecates while it eats. The feces are full of rickettsia. The bite itches. You scratch the bite.
And you scratch the poop into the wound.
You inoculate yourself with louse feces. That's the transmission.
Charming. Now, we usually associate this with history books or refugee camps. But there is a reservoir in the United States that is famously weird.
The southern flying squirrel.
I still can't get over this. Flying squirrels.
Glaucomys volans. In the eastern U.S., these cute little nocturnal gliders carry a strain of R. prowazekii. They have their own lice and fleas. Occasionally in the winter, they nest in attics. People inhale the dust from the nests or come into contact with the ectoparasites.
So you can get epidemic typhus in a suburban house in Virginia.
You can. It's rare, but it definitely happens.
How does the typhus rash differ from the RMSF rash? This is a classic exam contrast.
It is the directional opposite. Remember, RMSF is centripetal, moving inward. Typhus is centrifugal, moving outward. It starts on the trunk, like the axilla and chest, and spreads out towards the limbs.
Okay, so RMSF is wrists to trunk. Typhus is trunk to limbs.
And importantly, typhus usually spares the palms, soles, and face. RMSF hits the palms and soles.
Now, what is Brill-Zinsser disease?
That is the sleeper cell phenomenon. R. prowazekii can go dormant in the lymph nodes after the patient recovers. It can hide there for decades.
And then what?
Then, maybe 40 years later, the immune system weakens. Due to age, stress, and other illness, the bacteria wake up and cause a recurrent episode of typhus.
Without any lice involved.
No lice needed for the flare-up. But here's the danger. If that person has lice now, the lice can pick it up and start a new epidemic. This is how typhus persists between wars. It hides in the survivors.
That's incredible. Let's briefly mention scrub typhus. This is a massive global issue.
Huge. Ehrlichia tsutsugamushi. It's found in the Tsutsugamushi Triangle. Japan, Southeast Asia, Northern Australia, the Pacific Islands.
And the vector?
Chiggers. The larval stage of a mite.
Symptoms?
Similar to the others. Fever, headache, rash. But, like rickettsial, it often has an Escher. If you have a patient returning from a jungle trek in Thailand with a fever and a black scab, think scrub typhus.
Also endemic murine typhus, right? Just to cover all our bases.
Yes, Rickettsia typhus.
Yeah.
Transmitted by fleas, usually the rat flea or cat flea, found in Gulf states like Texas or in California. Milder than epidemic typhus, but same idea.
Okay, let's leave the vasculitis makers behind. We are moving to section 5, the anaplasmatacea, the white blood cell invaders.
We are shifting strategies. We are leaving the jailbreak and entering the bunker. These bacteria infect leukocytes, your immune defense cells, and stay inside the phagosome.
There are two main players here, Ehrlichia and Anaplasma. They are clinically very similar, but they target different cells. How do we keep them straight for the exams?
The names tell you the target if you know the full species names, but the easiest mnemonic is MEGA, M-E-E-J.
Here, Rick, meet me down.
M is for monocytes, which pairs with E for Ehrlichia. G is for granulocytes, which pairs with A for anaplasma.
Simple. Ehrlichia hits the monocytes, Anaplasma hits the granulocytes, usually neutrophils.
Correct. And visually, this leads to a very specific finding on a blood smear. Because they replicate inside the vacuole, the bunker, they form a cluster, a microcolony.
And it looks like a blackberry or a mulberry.
Exactly. We call it a morulae, which is Latin for mulberry. If you look at a blood smear and see a big purple blob inside a white blood cell, that is the morula. That is diagnostic.
Let's talk about the specific diseases. First, human monocytic ehrlichiosis, HME.
Caused by Ehrlichia chafensis. The vector here is the Lone Star tick, Amblyoma americanum.
The Lone Star tick, that's the aggressive one, right? The one that actively hunts you down.
Very aggressive. It has a distinctive white dot on its back. It's found in the southeast and south-central U.S., essentially the same map as RMSF. The primary reservoir is the white-tailed deer.
What are the symptoms of HME?
It's often called spotless Rocky Mountain spotted fever. You get the high fever, the headache, the muscle pain. But you rarely get a rash in adults. Kids sometimes get one, but it's not the rule.
And because they're blowing up your white blood cells, the labs look different than RMSF.
Right. With RMSF, the white count is often normal. With ehrlichiosis, you get leukopenia, which is low white cells, and thrombocytopenia, low platelets.
And liver enzymes.
Often elevated. So the triad of fever plus low WBC and platelets plus high LFTs in the summer in the south. you should be thinking Ehrlichia,
Now, human granulocytic anaplasmosis, HGA.
caused by Anaplasma phagocytophilum.
Yeah.
It targets neutrophils. The vector is the Ixodes tick, the black-legged tick.
Ixodes, wait, that's the Lyme disease tick?
It is, and that changes the geography entirely. Anaplasma is found where Lyme is found, the upper Midwest, like Wisconsin and Minnesota, and the Northeast, like Connecticut and New York.
Since they use the exact same tick, can you get both at the same time?
You absolutely can. We see co-infections. You can get Lyme, caused by Borrelia, and anaplasmosis from a single tick bite.
That sounds like a diagnostic nightmare.
It is. If a patient with Lyme is treated with doxycycline, it covers both. But if you treat Lyme with amoxicillin, which works for Lyme but not anaplasma, the patient might stay sick because you missed the co-infection.
That is a high-yield clinical pearl for everyone listening. If a Lyme patient isn't getting better on amoxicillin, you have to check for anaplasma.
Exactly.
Treatment for both Ehrlichia and Anaplasma.
Doxycycline.
Yeah.
Still the champion. Always the answer here.
Okay, we have covered the gel breakers and the bunker builders. Now let's talk about the outlier, the acid lover, Section 6, Coxiella.
Coxiella burnetii, the cause of Q fever.
Why Q?
It stands for query. When it was first described in slaughterhouse workers in Australia in the 1930s, the cause was unknown. It was a query fever, and the name just stuck.
Coxiella is the survivalist of the group. It is the only one that isn't strictly arthropod-borne in humans.
Right. While ticks do spread it between animals, humans almost always get it via inhalation. It is an aerosol disease.
And this is because of its structure. It forms a spore-like variant.
Yes, it forms a small cell variant that is incredibly durable. It resists heat, drying, sunlight, and common disinfectants. It can survive in soil or dried feces for months or even years.
This makes it a potential bioterrorism agent, doesn't it?
It does. It's categorized as a select agent because
Yeah.
you can aerosolize it. It's highly infectious, honestly. A single bacterium can cause disease, and it travels miles on the wind.
So where do people catch it naturally?
Barnyard dust. Coxiella concentrates in the placenta and amniotic fluid of infected ruminants. Sheep, goats, cattle.
So a goat gives birth?
The fluids contaminate the soil dries out, the wind picks up the dust containing these spore-like variants, a farmer or a vet breathes it in.
And then it gets into the macrophage and does its acid lover trick.
Right. It wakes up in the phagocytosome.
Let's talk about the disease itself. It has two phases, acute and chronic.
Acute Q fever is usually a self-limited flu-like illness. High fever, headache behind the eyes, and often hepatitis or pneumonia.
So atypical pneumonia in a farmer.
Yes. But chronic Q fever is the killer. This typically happens in people with pre-existing heart valve disease or immunosuppression.
And it causes endocarditis.
Culture-negative endocarditis.
Why culture negative?
Because you can't grow Coxiella on routine blood agar plates in the lab. If you have a patient with a heart murmur, vegetation's on the valve on echo, but the blood cultures are repeatedly sterile, you have to ask about farm exposure and test for Q fever.
Now, this brings us to what I think is the most confusing part of this chapter for students, the serology. Phase I versus phase II antigens. Let's clear this up.
It is tricky, but let's simplify it. Think of phase 1 as the wild-type full armor. It has the complete LPS structure. It is highly infectious.
Phase I is the real deal.
Right. Phase 2 is the lab weakling. After you pass the bacteria through cell cultures in a lab, it loses part of its LPS. It becomes truncated and virulent.
Okay, so how does knowing about these antibodies help us distinguish acute from chronic infection in a patient?
In acute infection, the body reacts first to the exposed truncated parts. So you see high levels of anti-phase 2 antibodies.
Acute equals high phase II.
In chronic infection, the bacteria have been hiding there a long time. The immune system is finally ramped up to recognize the full armor. You see very high titers of anti-phase antibodies.
So if I see high phase I, it means the bug has been there a long time. It's chronic.
Exactly. High phase I equals chronic endocarditis.
And the treatment for chronic Q fever is intense.
It is. You use doxycycline, of course.
Yeah.
But you add hydroxychloroquine.
Why hydroxychloroquine?
This is pure mechanism-based pharmacology. Remember, Coxiella needs an acidic pH to replicate. Hydroxychloroquine is a weak base. It accumulates in the lysosomes and raises the pH. It alkalizes the vacuole.
So it basically neutralizes the acid bath.
It turns off the activation switch. It stops the bacterial growth and makes the doxycycline work better.
How long do they have to take this combo?
18 months, sometimes longer. It is a massive commitment.
Wow. Okay, we have covered the entire map. We've gone from the actin-surfing Rickettsia to the bunker-hiding Ehrlichia to the acid-loving Coxiella. Let's lock this in with Section 7, rapid recall.
I'm ready. Let's do it.
Okay, I'm going to give you a scenario. I want you to walk me through your thought process and give me the diagnosis. Question 1. A 25-year-old man in North Carolina goes to the ER in July. He has a fever of 104, a severe headache, and muscle pain. He remembers pulling a tick off his leg 5 days ago. The doctor notes a rash starting on his wrists and ankles, spreading to his chest.
Okay, North Carolina in the summer plus a tick bite puts me right in the vector-borne zone. The key here is the rash pattern. Centripetal starts on wrists and ankles and moves in. That is the hallmark of Rocky Mountain spotted fever, or Rickettsia. The timing, 5 days post-tick, fits the incubation and reactivation period perfectly. Treat immediately with doxycycline.
Question 2. A 50-year-old veterinarian presents with fatigue and weight loss over 3 months. He has a new heart murmur. Blood cultures are negative. He mentions helping deliver lambs on a farm earlier that spring. Labs show extremely high titers of phase I IgG antibodies.
The exposure to sheet birth products, the placenta, screams Q fever, Coxiella. The heart murmur and negative cultures suggest culture-negative endocarditis. The duration of 3 months and the high phase I antibodies confirm this is chronic Q fever. He needs doxycycline plus hydroxychloroquine.
Question 3. A hiker in Connecticut comes in with fever and chills. He found a small tick on him last week. He has no rash. A CBC shows low white blood cells and low platelets. A peripheral blood smear reveals a mulberry-shaped inclusion inside a neutrophil.
Connecticut means lime country, but the mulberry inside a neutrophil is the clincher, that is a morula. Since it's a neutrophil, which is a granulocyte, this is human granulocytic anaplasmosis, Anaplasma phagocytophilum. The vector was likely an ixodes tick.
Question 4. A woman living in an apartment building in Boston develops a fever and a rash that looks like chickenpox vesicles. She has a single black crusty scab on her shoulder. She has seen mice in her apartment.
Urban setting plus mice equals rickettsial pox. The vector is the mite, Leponosoides. The rash is vesicular, pox-like, and the black scab is the Escher. Diagnosis is rickettsial pox caused by Rickettsia cari.
Final question. Question 5. A 6-year-old boy presents with high fever, petechio rash on palms and soles, and a history of a tick bite in Tennessee. You suspect RMSF. The parents are worried about antibiotics staining his teeth. What do you prescribe?
You prescribe doxycycline. It is the first-line treatment for RMSF in patients of all ages. The risk of dental staining is minimal with a short course, but the risk of death from untreated RMSF is extremely significant.
Boom, nailed it. If you listeners missed any of those, go back and review the summary of clinically significant organisms tables in Murray Chapter 34. It's a great cheat sheet for consolidation.
And really pay attention to the vector column in that table. Ticks versus lice versus fleas versus mites. In exam questions, the bug tells you the bug.
Before we sign off, leave us with a final thought. We've talked about a lot of mechanisms today. What's the one thing that sticks with you?
We spent a lot of time on details, but I want you to think about the elegance of Coxiella. Most pathogens spend massive amounts of energy trying to avoid the host's defenses, building capsules, secreting toxins to kill cells, stopping lysosomes. Coxiella does something entirely different. It takes the host's most potent weapon, the acidic lysosome meant to dissolve invaders, and uses it as a signal to wake up. It is essentially using the enemy's ammunition as fuel. It's a humbling reminder that in biology, every single defense mechanism creates a niche for something else to exploit.
That is a terrifyingly beautiful thought. The acid bath is the alarm clock.
Precisely.
Thank you all for listening to this deep dive into the stealth invaders. Stay curious, check your socks for ticks, and we'll see you on the next deep dive.